Thank you very much for providing me the opportunity to present my work on allelic variation in ACE2 and natural resistance against COVID-19. I would not like to go into the details of the SARS-CoV-2 and COVID-19, since most of you are being very well aware of the virus and the disease by now. So I would like to jump to start my presentation while talking briefly about the current status of the COVID-19. According to the estimates, as reported by the WHO, roughly around 23 million of the world population has been infected with the virus, out of which 800,000 individuals have died because of the disease amongst the closed cases. However, the morbidity rate, the mortality rate, and the transmissibility of the virus varies in different geographical regions, as represented in these maps, where the dark color areas represent the high morbidity, mortality, or transmissibility rate. That variation in the morbidity, mortality, and transmissibility depends on a variety of factors. For example, implementation of the preventive measures, which also varies in different countries, healthcare facilities, climatic and environmental conditions, age groups, as it has been clearly demonstrated by now that the pathological manifestation of the SARS-CoV-2 infection varies considerably among individuals of different ages, where in the old ages, the disease turned into a severe form in most cases, and with a high mortality rate. In addition to that, the genetic architecture of the general population of different countries could also influence the severity of the disease and may confer, in other words, resistance against the viral infection in certain individuals. So that genetic variations has been previously documented to confer resistance against or susceptibility against HIV, where the CCR5 or CD4 receptors being mutated, and in individuals with those mutated receptors may confer resistance or susceptibility against the HIV infection. And this forms the basis of the present work. In March 2020, co-crystal structure of the SARS-CoV-2 spike protein with the human receptor ACE2 has been reported. This, along with the previous studies, conclusively proved that SARS-CoV-2 uses human ACE2 as its primary receptor for the viral attachment. The ACE2 receptor over here is being represented with the purple ribbon, whereas the spike protein is being represented with the gold ribbon. The spike protein of the SARS-CoV-2 is roughly around 1,000 amino acid long protein, and out of which the region between the 438 to 506 amino acid, that small region, actually involves in the binding with the ACE2 receptor. And that particular region of the SARS-CoV-2 spike protein is referred to as receptor binding motif. Comparison of the receptor binding motif between the SARS-CoV-1 virus and SARS-CoV-2 shows that there's a considerable homology between those regions with some variations, and that variation may eventually lead possibly for the virus to cross the species barrier. So it may go from bat to pangolin to humans. The receptor binding motif of the SARS-CoV-2 spike protein interacts with the peptidase M2 domain, which is being present at the N-terminal of the ACE2 receptor. And that interaction mostly being confined to the N-terminal region between the RBM of the spike protein and the peptidase M2 domain of the ACE2 at the N-terminal region of the ACE2. Well, some of those interactions are being present at the central part of the domain. So now we focus on only those interaction points between the SARS-CoV-2 spike protein and ACE2 receptor. The Genome AD database shows there are more than 200 missense variants being present across the ACE2 protein. And out of which only 17 variants are being reported, which are being located at exactly the position where the ACE2 protein interacts with the SARS-CoV-2 spike protein. So then we focus our studies towards those particular variants. This table represents the list, shows the list of those variants, the 17 variants that are being located at the interaction points between the ACE2 protein and the SARS-CoV-2 spike protein. And the first column represents their accession number, and the corresponding amino acid change has been shown in the second column. So first we identified whether that mutation in the amino acid or mutation in the nucleotide, which in turn changes the amino acid, confer or encodes a protein which is being stable or unstable. So out of the 17 variants, 3 of those variants have been found to possibly encodes an unstable protein. So that may indicate that those variants, the protein encoded by those variants may be unstable and may confer some pathological consequences in the human, possibly. However, when we analyze all of those variants with 4 different programs that predicts the possible pathogenicity of the mutational variant, only one out of those 3 variants which encodes the unstable protein, only one variant, that is S19P, that has been found potentially detrimental by only 1 out of the 4, only one of the prediction program, that is polyphen-2. However, CEFT, CAD, and REVEL did not indicate any particular pathogenicity associated with this particular mutant. It is important to note here that there are several variations in the ACE2 that has already been associated with the hypertension, diabetes, and cardiovascular disorders. However, in the ClinVar database, none of those variants have been found to be associated with any human pathogenicity. I would like to also point your attention towards the allele frequency of those variants. Given the allele frequency is pretty low, so all of those alleles are being considered as a minor allele, which I would like to discuss later on in subsequent slides. So then we focus whether those structural variants, whether those amino acid change impose any kind of the structural change in the protein or not. For that purpose, we modeled all of those ACE2 variants, all those 17 ACE2 variants, and superimpose over each other. Once we superimpose over each other, from the ribbon diagram, it is very clear that all those ACE2 variants have a nearly identical structural topology as of the wild type protein. And their carbon alpha backbone deviation ranges from 0.17 degree angstrom to 0.58 degree angstrom. So given the structural topology is very identical, but when we compare the spatial orientation of the amino acid, of the substituted amino acid with the wild type amino acid, what we have found that spatial orientation of many of those substituted amino acid that is being represented here with purple color or purple sticks are different from the spatial orientation of the wild type residue that is being represented with the green sticks. That may eventually changes the intramolecular hydrogen bond pattern of the ACE2 receptor that may orient different kind of that may lead to the variation in the different kind of the interactions between the ACE2 protein and the SARS-CoV-2 spike protein. To explore this further or to dissect it further, we docked all ACE2 variants with the SARS-CoV-2 spike protein, and then compare the type of the interactions which are being present in between those molecules or in the complex. Here, the pink bar represent the charge-charge intermolecular interaction, which is one of the most strongest form of the intermolecular interactions. So as you can notice here, that most of those variants have less charge-charge interactions between the ACE2 protein and the SARS-CoV-2 spike protein compared to the wild type. It means that there is a possibility that those variants may weaken the interaction between the ACE2 protein and the spike protein and eventually may confer the resistance against the COVID-19 infection. But to further resolve it, we developed the intermolecular hydrogen bond or intermolecular interaction map. And in this connection, we have actually explored 3 different kinds of the interaction. That is the hydrogen bond interactions, electrostatic interactions, and hydrophobic interactions. And that results has been summarized in this table. So out of those 17 variants, there are 2 particular variants, that is the S19P and E329G lacks some of the very critical interactions which have been shown by the site-directed mutagenesis experiment that once been mutated, it abrogate the interaction between the spike protein and the ACE2 receptor. So in S19P and E329G, in both of those mutant, the interaction between the lysine 353 and glycine 496 and glycine 502 has been abrogated. That may indicate that these variants lack a critical interaction between the SARS-CoV-2 spike protein and the ACE2 receptor and may eventually confer the resistance. This has been further somehow strengthened. This notion has been further strengthened when we look at the binding affinity of the complex. So in the case of the S19P and E329G, the binding affinity is far less compared to the wild type binding affinity of the wild type complex. However, these results are computational and thus require experimental validation. Followed with this study, there are a few computational studies which have been conducted and reported later. For example, one of these studies suggests that N720D variant of ACE2, and that particular amino acid is not being located at the peptidase M2 domain rather than being located at the collectin domain, which is being located at the C-terminal portion of the ACE2. So that variant actually increased the affinity of the ACE2 to bind with the serine protease of the human, that is the TMPRSS2. Now, TMPRSS2 has been reported to prime the spike protein or to mature the spike protein by cleaving it into the quotient S1 and S1, S2. That is required for the invasion of the virus into the host cell. Similarly, it is also being required to cleave the ACE2 protein. So increased binding of the ACE2 with the TMPRSS2 may in turn increase the susceptibility of the individual which harbor that particular variant for the invasion of the virus, and so do the possibly the infection. Similarly, there are two preparental studies which also kind of support our conclusion, in which in one of these studies, S19P has been shown to have a protective role. However, there is another study which has been published in Journal of General Virology, where the variant which we reported to be possibly confer the resistance has been found possibly conferring the susceptibility. However, by and large, the data, the other studies also kind of support many of the conclusions which we have derived, or many of the inference which we have derived out of our studies. At the empirical side, there are 4 other studies which have been published or in the preprint stage. And 2 of them shows no association with the ACE2 variants with the SARS-CoV-2 infection or the COVID-19. However, one study linked it with the ACE2 expression, and the other study linked, which have been conducted in the Iran, suggests that 2 variants, that is the V485L and S331F, both of them are located at the peptidase M2 domain, may confer resistance or susceptibility against the COVID-19 infection, respectively. I would like to point here that most of the alleles, or all of the alleles which we have screened, or which these studies have actually screened, are the minor alleles. So it means they have a very less frequency in the general population. Thereby, any attempt or any investigation which is designed to establish any association or no association against the COVID-19 infection requires quite much of the technical and methodological and pathological stratification. At this moment of time, since the pandemic is in the very early stage, most of the research labs are being closed, and the availability of the samples are also linked with the health threats. So we can understand that those studies have been conducted generally on a very small cohort, and it warrants further investigation on a larger cohort size with a proper stratification, pathologically specifically speaking. And these have been highlighted, these points have been highlighted as well in the 2 earlier investigations by Kiddo and Bomba. So lastly, I would like to thank my postgraduate research students who worked tirelessly during this course and as well as counting right now as well. Nasiruddin here, Fozia, Sanya, Anusha, Aisha, Basma, and my colleague, Dr. Nusrat. Thank you very much for your attention.